reduction in terms of the unknowns, and in fact, putting together your unknown, God, I love all these cameras up here. Putting together your unknown charts, nitrate reduction actually had to take a part somewhere in the identification of the pseudomonads. Okay, so what reagents are added to detect nitrite? Just hear a little whisper. Alpha. Okay, naphtholamine and sulfonilic acid. Okay. Which tubes in this are positive for nitrate reduction to nitrite? So remember, when you do nitrate reduction, what's the first thing you look for? Gas, right? That's positive for nitrate reduction to? What's the gas? Nitrogen. Okay. If you don't have any gas, then you add the two reagents because you're looking for nitrite. If nitrite is present, what happens when you add the two reagents? So which one is positive for nitrite? B, the middle one. Okay. If you don't get a positive for nitrite, then you're looking for nitrate to see whether or not it's still left. It hasn't been reduced. Okay? What do you add to detect the nitrate?
Zinc.
Zinc. Okay. Which tube is positive for nitrate? If nitrate's present and then you add your zinc, what happens? If nitrate's still present, it's a negative nitrate reduction test. So after you add the zinc to your tube that hasn't changed colors with the two reagents, if nitrate was present, what happens? It turns red. Remember, nitrate is then reduced to nitrite, sort of artificial. If nitrate hasn't been reduced, it's still sitting in the tube. You add the zinc, it converts it to nitrite. You already have those two other reagents in there, and so it's going to turn red. So a positive test here, positive for nitrate, which means a negative nitrate reduction test, would be the red one. A positive for nitrate reduction at this point would be the yellow one. It means that it didn't take it to nitrogen gas, it didn't take it to nitrite, it took it to something in the middle. Okay. So this makes this, what is the center tube here? What is the center tube showing you?
Positive.
Positive for nitrogen gas. Okay? All right. This is a little bit more recent in all of your memory. This is BRBA. What components make it selective for gram-negative bacteria? What component makes it selective for gram-negative bacteria? Crystal violet and? The bile. They inhibit the gram-positive, making it selective for gram-negative bacteria. What component makes it differential for lactose-fermenting bacteria? Okay, the neutral red, which if there's lactose fermentation, the pH drops, and the neutral red will show the colonies as red, and a little bit of the bile may precipitate as well, but primarily it's the neutral red. It's the indicator dye that's present there. All right, don't count the colonies. What does SBCA stand for, and when is it used? Standard plate count. This was right off of your last quiz. And I would again look at your quizzes. Standard plate count augur. When is it used, or what is it used for?
To grow everything present.
A little louder.
To grow everything present.
Right. Well, aerobes and facultative organisms. It's not going to grow strict anaerobes, obviously, but aerobes and facultative organisms. It's a generalized medium. Don't count the colonies. But if there were 70 colonies on the plate, and that represented plating 1/10 of a mil of a 10 to the minus 3 dilution of a liquid sample, how many organisms were in the original sample? Okay, what's the total dilution there? It's 1/10 of a mil of a 10 to the minus 3 dilution. 10 to the negative 4. 10 to the minus 3, a tenth of that is 10 to the minus 4. That's the total dilution. Oops, sorry, that's on the bottom, 10 to the minus 4. 70 colonies and a 10 to the minus 4 dilution equals 70 times 10 to the 4th per mil. Okay? You better know how to do it from a liquid sample and from a solid, a food kind of sample. There'll be a fair number of points to put those together. Okay, what does MPN stand for? Most probable number. So you gotta get it all right, not most possible number, most probable number. What medium is used for this test? You guys have a lot of studying to do. What medium did we use for the MPN test? And why did we use it? What was that medium showing us?
Single strength lactose part.
We were looking for lactose fermenting organisms. The MPN test was what called what in the water quality test? Remember there were 3 parts? It was the presumptive test. We were doing the presumptive test for lactose fermenting organisms, okay? So we were using, well, lactose broth. We started with double strength because we were adding a lot. It makes it single strength, but it's lactose broth, okay? There's no indicator dye in this. How do we presume that we've seen lactose fermentation?
Gas in a Durham.
Gas in the Durham vial. And what gas is that? What gas from fermentation?
Carbon, CO2.
CO2. That's part of fermentation, okay? What medium is this and what is it used for? It's McConkey's, right? Because it's showing those little red colonies. So what is McConkey's agar used for?
Selectively culture gram-negative.
Gram-negative
That confirms
organisms, and then we're looking to distinguish coliforms from non-coliforms, lactose fermenters from non-lactose fermenters. So it inhibits gram-positive, right, allows the growth of gram-negative, and then it will allow you to see lactose fermentation on it. What's this medium? That's SIM, right? It's showing you the 3 things that SIM tests for. On the left, we're testing for motility, and that's just growth away from the stab line. In the center, we're testing for indole, and how do we do that? Kovacs reagent, and you'll see whether or not if indole's present, you'll get that red-pink color on the top. And the right-hand side,
hydrogen sulfide.
we're testing to see whether hydrogen sulfide is produced. What we're looking at is, what do we see here? The black precipitate, what is it? Iron sulfide. The organism produces hydrogen sulfide, there's iron in the medium that reacts with it, and we see the dark precipitate. Okay, so just be careful if it's asking what are we testing for, or what are you what is that black precipitate? Because there's a difference. We're not testing for iron sulfide. We're viewing iron sulfide to tell us whether or not the organism produces hydrogen sulfide. Okay, this is a giveaway kind of thing in terms of this plate. What's the medium? Blood agar, right? We have alpha, beta, gamma there. So what are the 3 results? What is the alpha? Partial what? Partial lysis of the red blood cells. Beta is complete lysis. And gamma? Non-hemolytic. Remember, this is hemolysis. Okay? Is the medium selective?
Differential.
Right, it's enriched and differential. Selective means that it keeps something from growing. This is enriched. It has the extra nutrients. Lots of things like to grow on it. Okay? And it's differential for? What does it differentiate between? You're looking at it. What does it differentiate between? Yeah, hemolysis. Whether or not an organism can do alpha, beta, or no hemolysis. That differentiates the organisms by the way they're growing on the medium. Okay? So if you're putting something on here and you're looking for beta hemolytic strep, right? Strep throat, you want to see beta hemolysis there. Okay, another sort of red pinky medium. What medium is this? Yeah, MSA, which stands for? Mannitol salt agar. Which organism is growing on the right? It's one of the few where you really need to know what the organisms do. Yeah, Staph aureus. Why is it yellow on the right? Acid from?
Mannitol fermentation.
From fermentation of mannitol. Okay? That makes it a differential medium. Organisms that can ferment mannitol will produce acid, changing the indicator dye, which is what? What's the indicator dye in this medium?
Phenol red.
Phenol red. Okay? So phenol red, somebody sent me an email this morning. I said we'd talk about the results. Who sent me that email today? Somebody was asking questions about phenol red, and I got this email this morning. I said we were gonna talk about it tonight. Nobody sent me that email, huh? Okay. So phenol red will turn yellow at pHs below about 6.8. You know, sort of a 6.8, 6.7, and maybe a little orangey. And some of you, I know when you were doing your carbohydrate test, going, I don't know, is this really acid, right? A little bit, but when it gets down, 6.6, 6.4, it will be yellow. If it goes above 8 or 8.4 or a little higher, it changes from sort of a red color to a more fuchsia kind of color, indicating an increase in pH. So that, again, it's an indicator dye. Okay, what component makes this medium selective? And what's it selective for? The salt makes it selective for halotolerant organisms, which is typically staph. There are a few other halotolerant, organisms, but it's primarily used for staph. And the component that makes it differential is there are only two components, guys. The mannitol along with the phenol red indicator dye. We're differentiating mannitol fermenting from mannitol non-fermenting organisms.
So if you just put halophiles or
It's
salt
more
tolerant,
halo, yeah, salt tolerant. Most of these are not halophiles, most of these are halotolerant. We don't really have halophiles on our bodies.
but you would have to put pseudomonas.
Pseudomonas.
Or what is that word? I forget what it's called. Staph, yeah.
Yeah,
So
Staphylococcus
staph is the answer.
is halotolerant, right. And it's really good because if you take a clinical specimen and you're trying to isolate out staph, most other organisms are not gonna grow because of the salt. Okay. Name the pathogen. Okay, does everybody sort of see? I have a little arrow here. Okay, this is not a gram stain, but this is not the best slide, which I'm pointing out here are endospores, an organism in a chain, endospores about the width of the organism. So that's indicative of, yeah, Bacillus anthracis, which causes anthrax. Okay? Okay, this one should be easier. What's this pathogen? What are we looking at here? So this is a smear that has a whole bunch of white blood cells on it. And we're looking at these little organisms that are inside the white blood cells, a clinical specimen taken from an inflammation, right? Yeah? Yeah. Okay, so it's Neisseria gonorrhea. This is really, really characteristic. It's in fact diagnostic. If you take a specimen and you look under the microscope, it's telling you. May not be the only thing you have, but at least it's telling you that you have Neisseria gonorrhea. What are the symptoms of the disease?
Discharge.
Yeah, a discharge. Okay. And how is it transmitted? Okay. STD, sexually transmitted disease. Okay, what's this pathogen? Treponema pallidum, okay? And it causes syphilis, And the shape of the organism is called?
Programming spider.
It's a spirochete, right? You have two spirochetes from the organism that you've looked at. Syphilis, the sample will be in a tissue. Borrelia burgdorferi, the sample will be a blood sample. Okay, otherwise you can't tell one from the other. They're just the spirochete. All right, this is a little tougher. So tell me what you see. What is this organism? Shape, gram reaction. Do you see endospores here? Anybody? No? It's a gram-positive. Yeah, bacillus abroad. Okay? So what's the organism? Doesn't form endospores. I told you there was only one that looks like this. Corynebacterium diphtheriae. Okay? Where is it found in the body? Okay, the back of the throat. Right? It produces a toxin that circulates around the body, and that's what causes the damage. So the organism lodges in the back of the throat. It's the only gram-positive, non-endospore forming rod that you had in that collection. Right. All right. So, first describe to me what you see here. Okay, yeah?
That's the tetanus one.
Yeah. What's the tetanus one called?
The
The tetanus one is not gonna cut it on the, on the practicum.
Clostridium tetani.
Clostridium tetani, okay, where is it found in the body? Yeah, it's usually in a wound, okay? This one should be easy right now. Okay, there's blood cells, a spirochete, right? Borrelia burgdorferi, and you better get these spellings right. And the disease is? Lyme disease. And how's it transmitted? Bite of a deer tick. Okay. What is this organism?
Clostridium botulinum.
Why do you know that this one's botulinum?
Because it's not at the end.
Right, so you can see that this is not quite totally at the end. There's some stuff here, here. It's close to the end, but it's not quite at the end. One of the things on the practicum is we'll go through the slides and then I'll go through them a second time. Because what's gonna happen is if you see a slide, you say, oh, that's Clostridium botulinum. And then 4 slides later you say, oh, that looks like Clostridium botulinum. You know, because they are difficult. And I've tried to pick slides that, you know, really are characteristic of what we're talking about, because sometimes it's really hard to see. Now, obviously, in a clinical setting, you know, from symptoms and from everything else that's going on, you're not going to say it's botulinum when it's tetani. So, where is the spore location? What's that called when it's not totally at the end? What's it called if it's at the end? Terminal. So what's it called when it's not quite at the end?
Subterminal.
Subterminal. Okay. If I said, where is the organism found in the body? What would you tell me? No, the organism goes right through the body. It doesn't establish. Remember, in an adult? So you are ingesting the toxin. So in actuality, you probably wouldn't even isolate the organism from an individual. You might isolate the organism from the food and be able to show that it's in the food. But it's basically an intoxication because the organism will go right through. The vegetative cells won't make it through. The endospores that are present won't be able to establish if you have a normal microbiome, and so you won't see that. Okay?
So wait, what was the answer?
You don't find it in the body.
Oh.
It's an intoxication. Okay, what's this staining procedure? That's an what's the primary stain? Allocated Okay, green, the decolorizer, water. Go back to, you have that quiz from early on in the beginning of the semester where I had you compare all through. The counter stain, safranin. Okay, don't name the organism, I don't think you can. But I could say, name a genus, not E. coli. Two genera that give you endospores. Both of them you should know because they were both on the list of organisms you looked at for endospores. One is
Clostridium.
Clostridium, and the other one is Bacillus. You had Bacillus anthracis, right? And you had the two Clostridia. Those are the genera that are endospore formers. Okay. What is this medium? Simmons citrate, okay. What is the dye called? The indicator dye? Bromthymol blue. And why does it turn blue?
Increased pH.
Yeah, because the organisms using the citrate cause an increase in pH, and that particular dye, when the pH goes up, turns blue. What test is this? Yeah, how do you know it's an indol test? Because the color, the reagents stay on top, okay? What's it testing for? Sorry. Sorry, let me just, I didn't turn this off. What the heck is going on? I don't know how to turn this off. Okay. Hello. It's not a phone call, that's why. Okay, I'll get it later. Sorry about that. Okay, so what is it testing for? It's testing for indole, but what is that testing for? If indole's present.
Tryptophanase.
If the organism can utilize tryptophan as a carbon source, if the organism produces tryptophanase, that's the enzyme if I ask you what enzyme, okay? And the reagent that's used, Kovacs reagent. Okay. I don't think I have a slide for the methyl red test, or the BP test. You can have the same kinds of questions about them, right? Okay, since a lot of you were still doing this for regular unknown, the carbohydrate fermentation tests are possible. The arrow is pointing to? Well, it's a Durham vial, and in that Durham vial is? What gas? CO2. Fermentation is always the CO2. The dye in the medium is? We just went through this with an MSA plate. The dye in the medium is? Phenol red, and the medium is yellow because? The pH has fallen. Okay. Okay. Oh, I do have a BP test in here. So what does the BP test for? Well, you're testing for acetoin, but what is that telling you? Non-acidic byproducts of glucose fermentation. Okay? Which tube is a positive test? Which tube is a positive test? Red.
Red.
The only time the red is a negative test is on the final stages of the nitrate test, right? That's the one that's sort of, usually red means positive. What reagents are added to the broth? KOH and alpha naphthol.
So if you were to put butadiene fermentation, would that be okay?
Yeah. All right. Which tubes are positive for glucose fermentation?
5, 4, 8, 3, 2.
Yeah, 2, 3, 4, 4A, and 5. Okay? Which tubes are positive for lactose fermentation?
2, 4, 8.
Which tubes? For lactose fermentation.
Oh, 4, 5, 4, 8.
4, 4A, and 5. Okay? Lactose fermentation, you'll get yellow throughout. Okay? Why do you streak the slant and stab the butt? Why do you do both?
What's glucose and what's lactose?
No, there's glucose and lactose throughout. But it does let you distinguish between the two of them. It's the same reason you have a tenth of the concentration of glucose to lactose. All of those things, and while you do it at 48 hours, all of those go together in distinguishing glucose from lactose fermentation. Because, yeah,
I'm sorry, exactly why is it that we have to wait 48 hours?
because what happens is when you, first of all, you stab the butt so that this is going to be anaerobic growth. And the slant is gonna be able to give you aerobic growth as well as anaerobic growth. So when the organism is inoculated, it begins, if it can use the glucose, it begins to use the glucose. If you remember way back when from lecture, when we had catabolite repression, organisms use glucose before they use lactose and don't turn on the genes for lactose until the glucose runs out. So they begin to use the glucose. And by using the glucose, they're gonna produce acid and they're gonna turn the tube yellow. There is a reduced amount of glucose present. If you read it too early, it'll look all yellow and you'll think, okay, fine, it's used everything. When it runs out of the glucose, if it can use the lactose, it'll turn on those genes, right? And there's a lot more lactose than glucose. So then it'll use the lactose and it will, again, have the pH low through lactose fermentation and it'll be yellow throughout. If it cannot use the lactose, the tube that was yellow from the glucose, in the aerobic region, it will use an aerobic metabolism that will use some of the components, other components in the slant, and as a result, it will produce ammonia that will increase the pH, and the yellow will then turn to red, which is why after 40, but it takes time for that reaction to occur, so that you look at it after 48 hours. In the butt, you're not gonna have that happening because it's anaerobic down there. So only up in the slant region will that occur, okay? So if you read it too soon, it could look totally yellow and you would think that it used everything. If you read it too late, it might even use up all the lactose and actually revert back to using some of the other components and switch the top. So it's designed to be read at 48 hours.
So just a question, so if you read it too late, the slant will be red, but the butt will still be yellow?
The butt will always remain yellow once the
Because
butt
of
is
the
yellow,
anaerobic.
because it's anaerobic and nothing else will happen down there. Okay, what's this test? Kirby Bauer. How do you know which antibiotic is most useful? What about the zone of inhibition? Will B and G, which have pretty much the same zone size, will B and G be equally as useful? By looking at the plate, can you tell that B and G are gonna be equally as useful? Well, I got every choice there. So, yeah, okay. How do you know? We just went over this in lecture. You have to compare it to that SIR chart, right? Because you saw there were different size zones meant different things depending upon the antibiotic. So you can't say that they're beginning equally as useful. One of them may be in the intermediate range, let's say, and the other one could be in the S range, and that'll be just fine. You always have to go to the chart. You can't look at this and say, E is the way to go. Now you can say A and C are out of the running because there's absolutely no zone associated with them. That's easy interpretation. But you really have to go and look at the chart. The chart we gave you is even less, is less complex than the actual chart that you sometimes get, because there are different zone sizes for different bacteria or for different sites of infection. In some cases, with certain antibiotics, like I say, go to the bladder and they're great for the bladder, but the zone size for the blood may not be as good. But that's why you go to the chart. Alright? And that's it for the review.